Thanks everyone for coming to the ESE PhD student colloquium. Today's speaker is Cassiano Becker. He's a PhD student here at Penn, working with Victor Preciado. Before coming to Penn, he did his undergraduate degree at Federal University in Brazil, and his master's at University College London. So he's going to be talking to us about spectral analysis of brain networks. So, Cassiano, you have the floor.
Thanks, and thank you all for coming. I'm going to talk about research that we did in collaboration with Sergio Piquito, Professor George Papas, Danielle Bassett, and Victor Preciado here at Penn, in collaboration with Professor Michael Miller and Scott Grafton at University of California, Santa Barbara. And the talk is about spectral mapping of resting functional connectivity in brain networks. That is a research problem, an open question, very actively studied in network neuroscience. And so I'm going to talk about network neuroscience to set the background. So network neuroscience represents and analyzes the brain as a network so that you can have an encoding of the complex interactions that arise in that system. And being a field of neuroscience somewhat recent, it has already enabled or enhanced a few important applications in the field, such as the diagnostics of disease, so looking at metrics of the networks, you can have earlier detection of some illnesses such as Alzheimer or other psychiatric conditions. Network neuroscience is also studied as a form of objectively measuring variables in cognitive science, for example, intelligence, learning, and development. It's also applied to provide an assessment of trauma, or how, if you affect physically your brain, how does that impact on your function of your on the dynamic functionality of the brain. And more recently, it's been also applied to brain-computer interfaces because it enables an encoding, an additional layer of encoding of the signals in your brain. The brain being a very complex system, you can look at it, a large-scale system, you can look at different scales. In the micro scale, you have the neuron as a unit, but you can also look at the mesoscale, where you look at the brain as a whole. And you also have the structural and functional views of the brain being represented as different networks. And that's behind, or that underlies our question here. So in the functional view, we're concerned about the dynamic patterns of our neuroactivity. Neuroactivity being the activation of neurons. They perform the action potentials, they impact on the other neurons, and we would like to see how does that behave in our brain as we perform different functions. In the functional view, the concept of networks is typically defined as a pairwise similarity between different regions of your brain. You can find that similarity to be in the time domain, in the frequency domain, as a coherence between two regions. And you can get this data through different physical measurements. For example, EEG and MEG, they measure directly the electromagnetic fields, and by doing so, you have a very high resolution. In time, you have a very fine scale measurement in time, but not so much in space, because the signals get a little scrambled. In fMRI, on the other hand, you have a very fine detail on the spatial scale, but not so much on the time scale. And also in the functional view, you have two different experimental paradigms, one of them being the REST paradigm, where the subject, the person, is asked not to do anything in specific, and you also have the task experimental paradigm, where you have very specific tasks that a person is asked to do. So typically the task is more studied in cognitive science, because you want to see how different people, by doing different tasks, how the signals are different in being measured in the brain. And REST, you typically, it's more studied for illnesses, because you want to isolate variables, for example. And yes, so these two experimental paradigms are studied. Of course, there is, you can also look at tasks for some illnesses which are related to cognitive ability. In the structural view, you are more interested in looking at the interconnection between different cortical regions of your brain, and then the networks. So you have the axons. Most of the connections in your brain are local, but some of them, and very importantly, you have interconnection between different remote regions, and those interconnections are believed to be important for the whole diversity of behavior that we are able to display. So you can define a network, a structural network, by quantifying how many of those fiber tracks are going from one direction to the other. And they are typically assumed to be static in short periods, although more recently people are also looking at potential changes but they are much smaller scale, but this is recently being studied. And you can measure it also, both fMRI and the functional measurements that I mentioned, and the structural, very importantly, they can be measured non-invasively. And the technology here is diffusion MRI, DTIs, diffusion, tensor imaging, diffusion, spectral imaging are some specialized ways of processing the diffusion measurements. And after you get that, you typically perform a tractography, which is you have to trace those vectors that you get here to reassemble the tracts and arrive at pictures like this or measurements like this. So just to get a view of how do you go from the instrument, the measurement, to the matrices. Both types of measurements, fMRI and dMRI, they are specializations of MRI. In fMRI, you're actually measuring blood and oxygen differences which occur in response to neuroactivation. So when your brain is functioning, the neurons are firing, they need glucose and oxygen to work, and that causes difference in the oxygenation of your blood. And the hemoglobin, which is in the blood, has magnetic properties that are differently sensed if your blood is oxygenated or not. So this is essentially the signal that's measured in fMRI. You get the time series. The time series, you get one potential time series per voxel, but then you assume a parcellation, which is a segmentation of the surface of your brain, and within that, each region of that parcellation, you average that signal, and then you do the pairwise comparisons to define your functional connectivity matrix. Whereas the diffusion MRI, you apply different angles of the magnetic field, and you see how the water in your brain diffuses with respect to those angles, and that tells you the preferred directions of water in the axons, and by tracing those preferred directions, you reconstruct an estimate of the axons in your brain, and then given the same parcellation, you have you count the number of fibers, or you average other geometric characteristics on those fibers to arrive at what we call a structural graph, a structural connectivity. And now our problem in specific starts from here. It is related to, given a structural matrix, can we find a mapping that brings our structural matrix to the observation that to another matrix? We call it F hat, which is an estimator or an approximation of the functional matrix that we measured. So this is the project that we are addressing here, the problem. There have been existing approaches. Typically, they started looking at direct correlations. So if you scope a few specific nodes, you can detect that there is a natural correlation between structural and functional matrices. The other approaches, people have built a simulation, dynamic systems-based simulations, which are informed by the parameters of the structural matrix. They simulate that system. That system generates a dynamic response. You compare then the similarities. You build a functional matrix out of that response, and then compare it to the original, and expect that these are close. And the other approaches, I look at some graph theoretical characteristics of the structural matrix, and I try to use those as predictors of the functional. The nodes, yes, the nodes are defined by the parcellation. And then you can have different notions of the edges, which are, you assume them beforehand. So in the structural, typically is the density of fibers. And in the functional, you use the same notion of nodes, but then you do a similarity between the time series of those nodes. And our proposal for that problem is, I will describe our functional connectivity as a mapping acting directly on the structural matrix. Can we define a functional form that transforms our matrix S to F? How would that look like? Can we describe it in terms of parameters? So that's the problem that we set out to address. And in doing so, we also wanted to see how it behaves empirically in the data sets that we have, both in different configurations, meaning we want to look at a specific individual. Can I, for that specific individual, provide a mapping? And also, can I find a characteristic that is similar across groups of individuals? So those are the two configurations that we intend to apply our method. So the idea behind our method is describe this mapping as a functional mapping, matrix mapping, starting from the structural matrix. And we use this very known fact from graph theory that if you take the powers of a adjacency matrix, those powers will account for the number of paths of different lengths between every element of that matrix, meaning, for example, in the matrix, in the element Aij or Fij of my matrix, I will say that the strength of my functional association between these two nodes is described by a combination of the different walks that I have available between these two nodes. So we do that, and then we also perform a rotation in the coordinates or the eigen-modes of the structural matrix to the functional matrix. And by doing so, we're establishing a mapping that goes from S to F hat, where F hat is applied on S, having these two parameters, A being a vector of, or a set of these coefficients. Here it is written as a vector, and R is a rotation matrix. And starting from that functional form, we want to apply it to the individual mapping problem that we call, and the group mapping problem. So for the rest of the talk, we're going to look at how do we solve that problem in terms of the estimation of those two parameters, and that is the method, actually. And then we're going to apply it to a set of 84 individuals, which is the paired data set that we have, for which we have structural and functional matrices. We're going to do the same for the group mapping problem, and then we can discuss some future work. So starting with the individual mapping problem, if we depict it in sets, we have a data set here for structural matrices and the functional matrix. We're looking at each individual pair here, and we want to relate these two quantities. In the literature, typically also people started, when they started studying this problem, they would average these matrices to what they would call a representative individual, and then look for a mapping between these two. So mathematically, the mapping problem is the same. In here, if you work with these two matrices, typically this is not that much advised, because the averaging between individuals doesn't have a clear interpretation. And so we are actually we're considering each individual separately. And so our problem here is, given Fj and Sj, given this pair, we want to find what are these two parameters that describe our mapping, such that our mapping is close to our original functional matrix in the Frobenius or Euclidean distance notion, which is the quadratic distance. So this is our mathematical problem given our proposed approximation. And so if we write out the notion that we saw in the previous slide here, if we write it out, we're saying that our F hat is a combination of the powers of the structural matrix, a linear combination, on which we apply a rotation matrix. And to begin to analyze that, we start by taking the eigen-decomposition of S, so we can write that F of S as the function actually being applied on the diagonal matrix itself. And if we also perform the eigen-decomposition of the F matrix, we have F here, and then writing F as this form here, F of S plus the rotation, we can start analyzing our original matrix and our approximation. And this, now rewriting the problem, we have this problem in a first level of analysis. So we're trying to find the set of coefficients and the rotation matrix such that this norm is minimized. We have this constraint here, meaning that the rotation matrix should be orthogonal. And if we look at it, we see that this is a complex, computationally hard problem. It's not convex. Just by looking at this set of constraints here, actually we have here a n choose 2 quadratic equality constraints. And also here we have this composition of the R matrices and the A. So our approach is to decompose that problem.
How do you tell it's not convex?
The equality constraints here are quadratic equality constraints. So for each column of R, you have that they have to be either 0 if they are different or equal to 1 if they are, if I equals j. And that those are equality quadratic equality constraints, and that is a non-convex problem. So we decouple this problem, and we observe that actually when we look at the problem written this way, we see that the A's are very much related to the phi's here in a way that we can interpret this as the A's are approximating the eigenvalues of phi, and the R is acting on the matrix V to approximate U. So we're using that fact.
But short question, like, why do you need to do the rotation? Like, because
It's
you have node I and node J?
it's yes. So it's an additional parameter space that we use. So rotation, we interpret this as being an isometric transformation between the eigenmodes of the structural matrix to the functional matrix. So in essence, we are expressing the U, the eigenmodes of the functional matrix, as a linear combination of the eigenvectors of V. So it's an augmentation of the parameter space, but one that we see that is still interpretable, that should give us, because we want to use the mapping, the idea is to use the mapping as to give us additional information as much. I mean, you look at you have the F matrix, you have the S, but the mapping between those two can also be interesting to look at, can give us information. So yeah, that was the reason behind. Later, we restrict the rank of R to see what's the effect. So when we decouple this problem and looking at this notion, we can, here's just a depiction. Actually, so the A's, we are using them to align the spectrum of S to the spectrum of F. And the R matrix, we're using it to align, to bring the eigenvectors of S to the eigenvectors of F. So here, these are actual measurements. So this is the first eigenvector of S and F, the one associated with the largest eigenvalue. And so breaking that problem or decoupling it, we're going to decouple, but later we're going to analyze the suboptimality in doing so. We're going to do that in 2 slides. So first, if we decouple it and we look at the problem on the approximation of the eigenvalues, it becomes a linear, a least squares approximation problem, where the coefficients here are now, the parameters are the eigenvalues, and this is turns out to be a van der Monde matrix, and the solution is known for that, is the linear, is the pseudo inverse of L. So that's the step 1. On step 2, so we're analyzing the objective function. So we call phi hat the matrix, the diagonal matrix that contains our approximation of the eigenvalues of F given our optimal set of coefficients that we found on step 1. So to analyze that, we define this matrix delta phi, which is essentially the error in the approximation that we got from step 1, and we now place that in our objective value, and we can compare. So we write the phi hat here as now phi plus delta, and now we can analyze this in this case that helps us to analyze if, for example, if we have 0 approximation error in the approximation of the eigenvalues, this delta goes to 0, so we remain with just phi here, and then by comparison we see that u should be equal to RV, and then we get 0 approximation error. So assuming that and placing u equals RV here, we now see that if we now compare these two quantities, this phi is eliminated, we get to this objective value, and by the invariance of the Frobenius norm under unitary transformations, unitary matrices, we see that this objective value is just the Frobenius norm of the vector of the error in the approximation of the eigenvalues, which is just the Euclidean norm of that error. So by assuming R equals UV inverse or UVT, the residual that we get in our approximation is just the error that we got in the approximation of the eigenvalues. And if we see here, if we increase k, then we can have as small approximation error as we want. So put more compactly, our estimation algorithm, if we want to call it so, is described here. We do the decomposition of V and of S and F. We mount the van der Monde matrix. We find the vector of the coefficients. We find the rotation matrix, and we have the optimal parameters. And then we can describe our matrix F hat as this functional form. And then we can apply that to actual data set to see how it performs. So here we have, in each vertical line, we have the distribution for all of the 84 individuals of the correlation between our F hat and F, which is the correlation between the matrices that we want to approximate. So in red here is that distribution when we're given both matrices, and we see both matrices S and J. So we see that as K is increased, not only we get a higher correlation, but it also gets a very low spread, very low variance. And in blue here, what we tried, or we are displaying is I train or I do the estimation on a subset of the time samples, and I evaluate the correlation with a functional matrix which was derived from another subset of time samples. And here in the scatter plot, we're plotting for all of the nodes in that matrix. That matrix here has it's 90 dimensions, so we're having 90 different regions. So we're plotting for all the n choose 2 elements of that matrix, the actual versus the predicted or approximated elements in red and in blue. And we also tried it for a increased parcellation, meaning we're subdividing the surface of the cortex in 600 regions. And then we also get the same qualitative type of behavior, although it's a little more spread because we now are predicting a 600-dimension matrix that has it's in the order of hundreds of thousands elements. Hundreds, yeah.
You explained again what's the y-axis?
The y-axis here, so we have our original matrix F, we have our F hat, we do the correlation between these two matrices. So if we do the vac of this matrix and the vac of this matrix, subtract it from the average elements, we do the correlation between these two vectors.
But for the parameters you found to minimize the Frobenius norm.
Yes.
So here the product of the Frobenius norm might be more natural. The Frobenius norm between the actual F and the F that you produce.
Yes. So typically in the field, they analyze correlation. Yeah, that's we could do both. Yeah, but in general, in papers, they report the correlation. It's the problem of subtracting the mean or not. Yes.
What's the difference between left and right? Is it 600?
It's the parcellation. So in here we're dividing the brain in 90 regions.
Two different parcellations.
Yes, that's the AL 600.
So I guess the red one for the training result, the blue one for the testing result, right?
Yes.
In your algorithm, when you compute this rotation matrix, the rotation matrix depends on both U and V, right?
Yes.
And then how do you do the test? And once you train, how can you find U? Because U actually from the function.
So in here, the red is we get a time course, a time series for all the nodes in the parcellation for the functional matrix. We take half of those time samples and we build our F or in-sample functional matrix. The other half, we build the out-of-sample functional matrix. So in here, we are estimating it with the in-sample functional matrix, and we're testing it on the out-of-sample functional matrix.
No, that answer. What I'm saying, if you go back to 2 slides, yeah, okay. Right. So you put number 5. So what I'm saying, when you use the training data, try to get this R star.
Yes.
And this R star, that's U. U actually from the F, from F.
Yes.
So when you do the prediction, basically you want to predict F. In this case, you do not U at all. Right? You do not U.
So,
You
right.
do not F.
So in here, we use the U from the in-sample. We do use F and S.
Now the U, the example is from average?
It's a subset of the time samples of the resting state time sample. Am I going back? Oh, okay. And here we analyzed the correlation for each individual when we when we used different type of structural matrices. So here we use the connect type of structural matrix, which is essentially given by the density of fibers between regions, and FA is fractional anisotropy, is another variable which is typically measured in structural matrices that tells about the intensity of how, for each voxel, how much the non-isotropic was the diffusion. And FA is taken by the average of the FA of each voxel between these two regions. Maybe I went too much in detail here. And we do that to see if, okay, given what's the difference if we use different structural matrices, the results are qualitatively equivalent, but for FA we get a quicker or a less dependency on the number of paths. And we also then restrict the rank of the rotation matrix so as to restrict the number of parameters in our mapping. And we also see that even for low rank, for just rank 3 out of 90, we also get a good correlation between the two spaces. And then we extended this for the group mapping problem, which pictorially we can see we now split it in individuals. So we use a subset of the individuals, which we call in-sample, to find the mapping parameters, and then we can test it either on the individuals in-samples. This one should be the out-of-sample variant, which would have an arrow here. And then our problem now is similar, but with the important distinction here that our parameters are unique, are just, they don't vary with, they don't have the same cardinality as the number of individuals. So we're looking for a common set of coefficients and a common rotation matrix across the set of individuals. And we also do the same approach where we decompose the problem. And we can see, again, pictorially here, we have a set of individuals. We want to use the pairs of those matrices to get a common vector of coefficients and a common rotation matrix Q. So proceeding in a similar fashion, the approximation of the eigenvalues, we can now form the same least squares problem. We get here a van der Monde matrix, which now includes all of the subjects in the in-sample set, and the optimal vector of coefficients is found equivalently by the pseudo-inverse matrix. But now for the rotation is where things got interesting. The C, the sub-I here, sub-I is for the power, and J is for the individual. So our C here is the same for all of the individuals. And now, if we look at the rotation problem, it's hard. We cannot do the same procedure of decomposing the matrices and comparing the eigenvectors, because now we want to find a rotation that we're now calling Q, which simultaneously minimizes this difference. And there's at least as far as we know, there's no closed form solution for this problem. So we start with an iterative optimization on the primal. So to do so, we can expand this objective function here, which becomes this problem. We're actually then doing this minimization of the minus the trace. And again, this problem is non-convex. It has roughly n squared quadratic equality constraints. And then to do that, we'll do a optimization in the primal. And we're going to look at the duality gap. in the Primal, we adopted a set of algorithms that preserve the optimality constraint, the constraint at each iteration. So they are similar to the proximal methods, which are more widely known. But in this case, they do a two-step projection in the feasible set. So you define a tangent space to the constraint set. You do a step, you project on the tangent space, and then you have another projection from the tangent space to your feasible set. And then to analyze the duality gap, we developed the dual problem, which is now, this is the Lagrangian. We have the objective plus the constraints and the Lagrange multipliers now called S and T. We have one Lagrange multiplier for each of the pairs of Qi, Qj columns. And then to find the dual problem, we note that this term here in general can be written, if we do this change of variable here, we just vectorize Q. We can group S here, S and I are here, T and I are here, and then these other quadratic products here using this identity, we can describe them as this quadratic form here. And then to find the dual, we note that this term here is unbounded unless this term, this matrix here, which is actually, it's a fixed matrix here, is positive negative semi-definite. And in that case, Q, well, given this constraint, the optimal solution is setting Q to 0. Then we can write the dual problem. And then we can solve the dual problem. With the note that because of this constraint, our LMI here is of order n squared, which is a large problem. So these are some runs of the Primal solution for different sizes of the number of elements in the matrix. And we see that if we set a stopping criterion given the gap as a tolerance, it's been achieved empirically. We can observe that. So even if we remove or we reduce the tolerance of the gap, essentially our Primal optimization problem is achieving arbitrarily low duality gaps. And then we were happy with this optimization approach. Then we can summarize the algorithm. It's pretty similar to the one for the individual case. While now here instead, in the previous case, we had a closed form solution for the rotation. Here we have an iterative descent optimization. But once we have that, we can then proceed with the approximation given those parameters in the same way, mostly. And so here are the results over the data set for both parcellations. Correlations are lower than the ones on the individual case. And we were expecting that because now we're casting the problem so that there's one set of parameters that should map every individual in each set. But it's still high. One thing that I didn't note is that typically correlations that were being obtained in the literature in prior work, they were around 50% in the average case of the individual scenario. So we achieved increased correlations, significantly increased correlations, and even in the group problem, we're achieving competitive level of correlations. And so we were looking at this global or universal parameters.
So I have a question about the previous slide. So there, in that case, the y-axis is the average correlation, because you have correlation with different.
It's the individual correlation. For each individual in my in-sample, for example.
That's the part of the
Yeah.
And in this case, the estimation is all in sample in terms of subjects. I mean, it could be out of sample in terms of time steps.
Right.
But in terms of subjects, it's all in sample.
Yes.
Did you try?
Yeah, there should be a legend here. The yellow one is in sample, and the green one is out of sample.
Out of sample in terms of subjects or in terms of
Subjects. Now it's subjects. Yes. So we were looking for this common parameters, and then once we got them, we decided to plot the eigenvectors of the rotation matrix in the cortical surface. So for each node, which is each element in our each dimension of our matrix, for each eigenvector, we plot the intensity here. So here are the top 4 eigenvectors, meaning the eigenvectors associated with the 4 largest eigenvalues. They turned out to map to in the cognitive when the researchers, Danny, Michael, and Scott, they were very excited to see that the intensity of the eigenvectors mapped to known regions in the cognitive literature. So here we see that the motor region is activated, and here in the third eigenvector we see the visual region, and here we see the decision making or the frontal cortex region, and the first eigen-mode, it's understood to be just an average or a bias that if we think of these eigenvectors as PCA components, giving the directions of energy of the signal, this is a bias that would minimize the average energy between the eigenmodes. So we also did this the same plot for the higher parcellation, for the 600, and we saw that qualitatively the activated regions were quite similar. Now 600 regions is much more fine scale, so the activations are a little bit more located, but the results were equivalent. So in summary, looking at this problem, we proposed this mapping model and algorithms to estimate the parameters, which allowed us to have a high accuracy of correlation between structure and function connectivity in the individual level. It also enabled us to do this analysis at the group level, and it produced or enabled us to look at the eigenmodes as a source of information of the commonality between groups of individuals. And then as next steps, we now want to take that further, do a more probabilistic analysis in looking at the rank of the matrix, estimation characteristics of this mapping. We can think about learning the mapping parameters, meaning now if I have a mapping of S and the parameters, can I just, looking at S, can I learn the parameters so that I can just use the S matrix and then given the S matrix I can predict F. We can think about multimodal mapping, including other sources of information, analyze task data, think on the inverse direction of the mapping, and even apply this to other network domains. Yes, and these are some. So this research is described in this paper here in archive. Here are the reference on the with the optimization. Here is prior work in that problem. These two papers. Yes, and with that I close. They are weighted. They are weighted and thresholded. So typically for lower values, they are cut. Typically they are 20% dense. Yes.
And the weights, so when you start applying powers of this matrix, then you have a problem that they might amplify or attenuate the effect. So you have to sort of tune these weights somehow? Or?
Yes, so the weights are then found by that optimization. We could normalize, do Taylor series expansion where you have the factorial in the denominator. We could try to even them out. Yeah.
And then the coefficients of this polynomial that you find, the A's, are the coefficients decreasing somehow? Or something? Like you observe the actual ones that you get?
They are decreasing, but their significance is related to that normalization issue. So when we were analyzing the results, and then we wanted to give a notion of how much increasing powers contribute to the prediction, we decided to interpret those based on the incremental gain in the correlation, instead of looking at the magnitudes of the coefficients. Right. But as next steps, we want to look at the spread of the similarity between these coefficients. How can they be, for example, used as biomarkers for subsets of different populations, for example, under different conditions. So what we expect is that the mapping parameters themselves should give us relevant information. So yeah, it's linear, but then it's composed with the rotations. So it's not totally linear, but we could start thinking about kernels or yeah. The eigen, right. So the Q matrix now is both. It includes the rotation and the eigenvectors of the structural matrix. So the Q matrix that we're actually finding, we could think of that as the estimation, the best estimation of the eigenvectors of F. So that problem we could think of it as like a multi-subject PCA of the functional matrix.
So you mentioned that some of the results that you had are actually matching what the neuroscientists know about some functional
Right.
about different parts of the brain. So were there any results results that were significantly new or unexpected?
Unexpected. I think the fact itself was a little unexpected. But if you think about it, the eigenvectors, because they are orthogonal, they shouldn't be too similar within each other. And then you have the parcellation is self-discrete, so you have more definite regions which are constant in that region. So I think what's interesting there is that fact, for example, that the motor appears as the second eigenvector being the most relevant to explain the functional connectivity matrix. So for me, the fact that the cognitive regions appeared, it's kind of by the definition of the parcellation. Maybe I'm being like too strict here, but for me, I think that is even more interesting. That you have that ordering of the explained variance.
One more quick question. So, the functional matrix F is something that you computed from data?
Yes, yeah. It's defined as, you can define it in different ways by the correlation between the time series of each node. So you can do time correlation or you could do frequency correlation.
I wonder whether you could do something without going into finding the matrix F first and then trying to explain that matrix F, but somehow try to explain the original data in some kind of different representation. I don't think thought about that. Before summarizing, let's say, in sense data into the matrix.
You mean, I'm not sure I understood.
How much of the correlation is explained by the rotation matrix?
Right. Yeah, we can have a sense from here, I would say. So here, even for polynomial order K1, you have an initial correlation which is significant. So the rotation is really is part of the model. Yes, it's helping in achieving high correlations. Okay.
Why can't you use the eigenvectors of the already available functions?
We are using.
The figures that you show are the distribution of eigenvectors of function connectivity, not estimate.
You mean on the cortical surface, the plots that we did? Because those are, we somehow restricted them to be equal across all individuals. So those would be it's the representation of the functional eigenvectors, which is common across all the individuals. You could see if averaging gives similar results or not. Yes.
So, very interesting. And you have this two-step procedure. Which step is more important in the process? Let's say if you think you omit the first step, just compute the metric momentum between F and S. Just do the rotation. Have you tried? What have you done?
I haven't explicitly tried that. I think it's related to the question on that Amin did on the rotation. So if you restrict your k to be 1, you have an amount of correlation. Or if you restrict your rotation to be identity, you would also have another extreme case. So yes, but I haven't objectively done an experiment trying that.
So the second question is, when you deal with a big matrix such as 600 you have, do you have any kind of computational issue when you compute the least square?
Right. It was not so. The least squares problem. It was quick. It didn't take long. In the individual problem, no, I didn't have any computational issues. In the group problem, where we had to do the iterative primal optimization, the primal optimization was actually doable. It's just the dual bound. It's very hard because you get that LMI is of size n squared. So typically, then you can you will do only the primal optimization, and then you can have a sense of the gap, because you can set these objective values to be normalized between 0 and 1, and then you can have a sense of how suboptimal you are. So computationally, I would say it's the most demanding step is the dual problem in the group case.